Good afternoon, everybody. Thank you so much for joining us for today's Leaders Initiative seminar. My name is Stephanie, and I'll be monitoring things here on the back end. If you have any questions, want to chat, you can put any questions that you have in the chat or comments, and we'll be sure to address those as they come along. Our next Leaders Initiative seminar is going to be August 26th, so mark your calendars. Dr. Tong Tong Lee will be presenting brain affective connectivity analysis using causalized convergent cross mapping. Towards the end of this event, I will be sending out a Qualtrics evaluation, so please take the time to fill out the evaluation. We really value your opinions, and we want to see which kind of topics you are looking forward to learning about in the future. And with that, I'll turn things over to Courtney for the introduction.
Great. Thanks so much, Stephanie. And thanks everybody so much for joining us today. We're excited to welcome Dr. Sarah Elzinga as our speaker for this month. Dr. Elzinga is a postdoctoral fellow in the Department of Neurology at the University of Michigan, and she will soon be starting as an assistant professor in the Department of Physiology at Michigan State University. The goal of her research program is to understand how immune system pathways respond to damage at the cellular level and promote injury of the nervous system. She uses multiple models to examine this injury and understand how it might promote cognitive impairment. So now I'll turn it over to Dr. Elzinga.
All right, great. Well, thank you so much for that introduction, and thank you everyone who is joining us on this lovely Monday noon time meeting. I'm happy to be here and to present to the group a little bit about my work, as Courtney mentioned, looking at inflammation in the nervous system. And I'm hoping to convince you that it's one of the factors that underlies cognitive impairment in individuals with obesity and with metabolic dysfunction. Make sure I can advance my slides here. There we go. All right, just to give you a little bit of an idea of what I'll be speaking with you today. First, I will give you a brief background on obesity and diabetes and what their complications are, and particularly emphasizing what their complications are in the brain. Then I'll talk to you a little bit about how we're trying to identify mechanistic targets of interest using some data from the clinic, as well as some recent data that we've gotten in conjunction with the MADRC and the Brain Bank to look at some human transcriptomics data. Then I'll talk to you a little bit about how we're trying to understand these underlying immune and inflammatory mechanisms. Also looking at how this is changing over time. So what's changing early versus what's changing later. And then a particular inflammatory mechanism we're interested in exploring called CGAS sting. Then I'll sum it up with some future directions and some conclusions for the work. All right, well, why am I and so many others interested in obesity and diabetes? Well, as it turns out, they're becoming an increasing problem, not only in the United States, but really around the world. I'm just going to use the United States as an example. So you can see here from this map, what this is telling us is the approximate prevalence of both obesity and diabetes in the States. So what we're seeing here is a darker blue color indicates a greater prevalence of obesity, whereas a darker red color indicates a greater prevalence of diabetes. And of course, if you put blue and red together, you get purple. So an increased purple color is telling us that we have an increased prevalence of both of these disorders. And you can see in 2004 across the United States, the burden isn't too high. But if we jump forward 5 years, that changes quite dramatically. Few more years into 2014, and the map color has just changed quite dramatically. And then finally, with the more recent data in 2019, you can probably appreciate that we have a very high burden of both obesity and diabetes across the United States. But what does this really mean, and how does this tend to progress along a so-called metabolic continuum? What we really tend to see is that increasing amounts of obesity tends to lead to the increased dysregulation of both glucose and lipid metabolism, where we have an eventual trajectory that tends to culminate in frank type 2 diabetes, where we get little or no production of insulin from those pancreatic beta cells. Now, all along this continuum, we have an increased risk and an increased prevalence of these so-called diabetic complications. And I'll get into what I mean by that in a moment here. But really, why are we seeing this? What are the underlying changes in our society that are potentially causing this burden of obesity and diabetes? We really think it's due to two primary factors. both this increasing consumption of these highly energy-dense, palatable foods, as well as our increasingly sedentary lifestyles. I say this as we are all likely sitting at our desks watching a Zoom presentation. But let me just give you a little bit of a picture of what this looks like in terms of our food consumption. So if you look at this typical fast food meal represented in the 1960s, and then its corresponding meal in 2011. So across this span of years, this same exact meal amounts to 1,000 extra calories that we are consuming in that time frame. Now, all of this contributes to our burgeoning costs in terms of dollars. So this isn't just cost in terms of quality of life, but this also equates to a cost in terms of dollars on our healthcare system. In fact, if we look at just the cost of obesity alone, the CDC estimated in 2008, so this was quite a while ago, that obesity alone costs us around $147 billion, and that's billion with a B. Of course, then, if you add in all of the associated diabetic complications, which occur also in obesity alone, this further inflates these costs. Now, what do I mean by diabetic complications? Typically, when we discuss these, we're talking about multiple organ systems that are affected by obesity and diabetes, and these can range from things like affecting the eyes and diabetic retinopathy, or affecting the nerves and diabetic neuropathy. But increasingly, researchers are recognizing and clinicians are recognizing that obesity and diabetes also impact the brain in the form of promoting cognitive impairment. And this can range anywhere from a lower IQ all the way on up to frank dementias like Alzheimer's disease. And this not only impacts any one particular area of learning or memory, but it can impact all of these major cognitive domains. And this can include, like I said, learning and memory. So how do we recall things? Or executive functioning, how can we plan? Are we flexible in our thinking? Or language, how do we recall words? If we're trying to find a specific word, are we able to find that word that we're looking for? All of these things can be impacted in the type of cognitive impairment that we see in individuals with obesity and diabetes. Now, everybody in this meeting and on this meeting is probably aware that there is some normal decline in cognition with aging, but really what we're seeing is this above and beyond, where we're getting either mild cognitive impairments, moderate cognitive impairments, or actual dementias in these patients that impact their ability to live their day-to-day lives. Not only that, but as I mentioned, we're talking about a continuum of disease. So how is this changing as we're aging? And not only that, but when we look at the burden of obesity, diabetes, and aging together, this really makes cognitive impairment a huge problem. Because as you can see from these projections that were generated by the United Nations, the number of adults that are in that 65 years of age and older bracket, which is at a high risk for the development of cognitive impairment, is only expected to grow from 2020s to 2050. In fact, what we will see in about 2050 is 1 in 5 individuals will fall within that age bracket of 65 years of age and older, making it a really big problem. All right, so we know that this is a problem. We know this is a growing problem and expected to continue to grow. How can we look at data from the bench? And how can we look at some data from humans to help us understand what are the underlying mechanisms of this increased risk for cognitive impairment? Both we and others have shown that it's not just obesity or not just diabetes, but multiple components of these disorders that might be contributing to cognitive impairment. For example, you can see these hazard ratios that are increasing with different aspects of diabetes, such as poor glucose control, in other words, poor HbA1c, or a longer duration of diabetes. So if you've had diabetes for a longer amount of time. Not only that, but if we look at the odds ratio that's associated with the metabolic syndrome, which is a compilation of different diabetic risk factors, including obesity and including poor glucose control, you can see that that's really increasing the risk of these patients to progress from a mild cognitive impairment onto actual dementia, or in this case, Alzheimer's disease. But as I mentioned, we've also seen this in the clinic as well with our patients. And what we can see if we look at cognitive deficits is that it's not just obese patients with diabetes, but it's even obese patients that are normal glycemic or have normal glucose levels that also have an increased prevalence of these cognitive deficits. So it's a combination of both obesity and diabetes that appears to be driving this increased cognitive impairment burden. We also have some data from this really interesting group of patients in Arizona known as the Pima Indians. Now, why these patients are so interesting for us to look at is that they've been followed for a really long amount of time in terms of their metabolic and their different diabetes complications. Some of these patients have been followed as early as 1996, so for quite an extended period of time. And in this population, they tend to develop type 2 diabetes rather early, much earlier than the rest of the population, around 25 years of age, and this is in both males and in females. Now, while I mentioned that they've been followed for quite a long time in terms of their more traditional diabetic complications, such as neuropathy or nephropathy, we've also recently been looking into their cognitive deficits or potential cognitive deficits. And in fact, what we're seeing is that they also develop these cognitive deficits earlier in life, around 40 years of age, which again is quite a bit earlier than most of the rest of the population. And what we're really seeing is an association between diabetes duration and their episodic memory. And essentially what this is and how this is measured is the patients are shown a series of images on a screen. Those images are taken away. There's a latency, and then they're presented the images again, but in a jumbled up order. And they have to try to correctly remember the original order of the photographs. An inability to do so is indicative of this poorer episodic memory. And again, we see that the longer these patients have had diabetes, the poorer these outcomes are. Now, I will also mention at this point that episodic memory, along with executive functioning, are two of these major learning and memory domains that are known to decrease with an increasing age. So our next step was then to, okay, we know that this is happening in the patients, but what might be underlying this? And so as I mentioned, we did this in conjunction with the MEDRC and the Brain Bank. And we were very fortunate to get a hold of these 6 patient samples. These were hippocampal samples that we looked at. And we had 3 control patients and 3 patients with both type 2 diabetes and Alzheimer's disease. We performed both spatial transcriptomics on 2 of our patients and single-cell transcriptomics on all 6 of the patients. And I'll share with you the spatial transcriptomics results first. Now, the advantage to this particular bioinformatics analysis and this particular omics analysis is that we're able to understand gene expression at a spatial level. So you can see here, for example, we have all these different clusters of gene expression that cluster based on their similarity. So it's different similarity in terms of gene expression profiles. And then we can map that back onto the area of the brain in which they are identified. Now, the slight disadvantage to this particular technique is that it's based on spots, or you can tell kind of hopefully from this picture, each of these tiny little dots represents a different sample or a different sampling. And each of these spots can contain anywhere from 1 to 10 cells. So we're not getting a single cell resolution, but we do get that trade-off of the spatial resolution in these data. But what did we end up seeing? Well, on a whole, en masse, what we saw is perhaps as expected, and you can probably appreciate from the pictures here, that in our 80 patient, we had quite a decrease in neurons, to be expected, but we also had an increase in microglia. In fact, if we pulled this out and we looked at these data in terms of the different clusters, and we tried to identify what was changing in terms of the percentage of these clusters between the patients, and what do these clusters contain, we can see that in many of the clusters that had a high degree of change due to Alzheimer's disease also had a high percentage of glial cells. But we wanted to understand a little bit deeper what was going on in terms of these changes. And so what we did is we used this particular bioinformatics analysis known as CellChat. And what this allows us to do is it allows us to assess ligand receptor pairs between these clusters to understand potential changes in signaling. And what we see is we have a couple of clusters that are having a really high degree of change or really high either upregulation or downregulation of their communication due to Alzheimer's disease. And again, we found that many of these clusters had a high percentage of glial cells. It was great that we were seeing these changes, but what in terms of the communication is changing, right? So we wanted to know on a pathway level, on a biological pathway level, what is changing. And we're able to, again, use both this ligand receptor pair analysis, as well as a more traditional differentially expressed gene analysis, where we look at changes in gene expression and we look at the enrichment of the different biological pathways of those genes. And essentially, I know it's too small to see, but I'm going to highlight it for you. What we see in terms of changes due to AD are a lot of changes related to, of course, neurodegenerative diseases, such as changes in APP signaling or in Alzheimer's disease pathways. We also see changes in metabolic or diabetic related pathways, but really we saw a nice enrichment or a nice change in many inflammatory or immune system related pathways, such as changes in SPP1 signaling and changes in things like antigen processing and presentation in the brain. Having this information, we wanted to take a step back and we wanted to understand it on a more single cell level. So this was our single cell transcriptomics analysis of all 6 of our patients. We were able to identify the expected cell types, such as neurons, microglia, astrocytes, etc. And then we used that data to apply this same bioinformatics pathway to understand changes on a cellular level. And what we were able to see is that, again, glial cells, in particular microglia, appear to be having a large degree of change in their signaling in AD versus in controls. In particular, we saw an increase in communication between microglia and these non-glutamatergic neuroblasts, and an increase in communication between microglia and vascular endothelial cells. And when we went to understand what these microglial changes in both signaling and in biological pathway enrichment really look like and what they're telling us, we found, again, changes in things like learning and memory, so kind of relating back to that neurodegenerative phenotype, as well as, again, in the inflammation or the immune system side of things, with changes in things like complement and coagulation cascades and B-cell receptor signaling. So really all the data from our human samples that we have and the data for the clinic were telling us that we are getting early changes and changes that are related in cognition to diabetes duration, to obesity, et cetera. And when we look at our samples using our transcriptomic analysis, that many of these changes are inflammatory in nature and that they appear to be overly represented or driven potentially by microglia, so these immune cells that are resident in the brain. So that led us to our hypothesis that the microglia are playing a key role in promoting this cognitive impairment under conditions of obesity and diabetes. So knowing all of these data, our next step was to try and understand these underlying mechanisms by bringing them back to the bench. So our first step was to pick our model. So in our lab, we tend to use the high-fat diet-fed mouse. And what this animal is a C57 black 6 mouse, non-transgenic. And what we do is we feed them a diet. 60% of that diet is fat, and that fat is derived from lard. So it's a very highly saturated fat, which is considered to be the bad fat. And what happens with these animals is that we're able to really well capitulate what we see in clinics. So the animals become obese, they develop glucose intolerance, they become hyperinsulinemic and dyslipidemic. Typically what we see in the mice is more of a hypercholesterolemia. They also have inflammation or immune cell changes. What we see are changes in the peripheral circulating immune cells. We can see things like infiltration of the immune cells into adipocytes or into adipose tissue, and we see an increase in things like plasma cytokine concentrations. And then, of course, importantly for us, they also develop cognitive impairment. And we and others have consistently shown that this model does develop cognitive impairment both early and fairly consistently. But what does this look like over the lifespan of a mouse? And how can we really relate that back to our human data, which is really what we're interested in doing? Just to give you a brief snapshot of how we can look at a mouse in relation to a human in terms of aging. So a 5 or 6 week old mouse is essentially a teenager. We really don't see them reaching middle age until about a year old. And then they're considered older or aged around a year and a half, which if you look at that in and try and translate to that human years, it's about 56 years old in a human. So not terribly old, but older. So wanting to understand next, what's changing early, what's changing later, how are these changes impacting disease during the disease course? We first wanted to start with our acute model. So in order to do this, we took that C57 Black 6 mouse at 6 weeks of age, and we fed them either a standard diet or a high-fat diet for just 4 days with a glucose tolerance test on day 3. And surprising to us, we saw even at this very, very acute time point, after 3 days on diet, you can probably appreciate from this graph here, they were already glucose intolerant. We can see this on the curve here as well as in the area under the curve. And also, interestingly, when we looked at their brains after 4 days on diet, we saw that this critical insulin signaling protein, AKT, was no longer responsive to insulin stimulation, which is telling us that they're already developing signs of insulin resistance in their brains after 4 days on high-fat diet. But what about immune cell changes? What are we seeing in terms of their immune system? Well, in terms of in their periphery, so in their circulating blood, we saw an increase in cells that were more associated with the adaptive side of the immune system. So the more specific, slower to respond side of the immune system, where we saw an increase in B cells, an increase in T cells, and a slight decrease in Ly6C positive monocytes. But conversely, when we looked at the brain, we actually saw more changes in that innate immune side of the immune system, where we saw an increase in neutrophils, an increase in microglia, and an increase in microglial cell size. Additionally, we actually gave these animals lipopolysaccharide, or LPS, as a way to further challenge their immune system to ensure that their immune system was working as expected. And when we did this, we saw that in the brain, both the standard diet and the high-fat diet mice had an increase in the number of Ly6C positive monocytes in the brain in response to LPS. But when we looked at the expression of CD11C on these same monocytes, we actually saw a nice decrease in our standard diet mice given LPS. But our high-fat diet mice already had a pretty low expression just starting out, and LPS was no longer able to influence the expression of CD11C on these monocytes. So what is this really telling us? It's telling us that 4 days of high-fat diet is recruiting immune cells into the brain, innate immune cells, and it's potentially activating these cells and particularly maybe activating the microglia. But we really wanted to dig into this a little bit deeper because that's not really the best way to assess microglial activation. In order to do this, we applied this really interesting morphological technique where we can take pictures of the brain and we can assess the different morphological characteristics of our microglia. Now, this is important and interesting and gives us a lot of data because microglia are highly plastic cells that respond rapidly to their environment in order to change their function. So a more resting or homeostatic microglia is going to have these nice long processes. It's going to occupy a large 3-dimensional space. And it's more so just kind of hanging out, making sure everything's functioning as normal. However, when you activate these cells, they suck in those processes, so they become shorter, they have fewer branch points, and they occupy a smaller 3-dimensional space. And I would imagine you can probably appreciate from the picture here, in these standard diet mice given saline, you can probably appreciate the highly branched structure of these cells. But then again, when we challenge them using lipopolysaccharide or LPS, they pull those processes in and they can become more this amoeboid-like shape. Interestingly, when we look at our high-fat diet animals, we can see that their cells are already fairly activated in their morphology. And when we give them LPS, there's no change in response to that immune challenge in terms of their morphology, which you can probably appreciate by the quantification shown here on the right. Telling us that 4 days of high-fat diet feeding in our animal model is sufficient to activate our microglia to such a state that further challenge with LPS is unable to further alter their morphology. So after we saw these really interesting acute changes in our animals, we wanted to understand what was going on a more chronic scale. So what happens if we feed our animals high-fat diet for a full year? So we did exactly that. We started them at the same age, 6 weeks of age, so as teenagers, and we fed them for a full year until they were essentially middle-aged at a year old. During that time, we performed repeated assessments of their metabolic phenotype using glucose tolerance tests, as well as repeated assessments of their cognition using different cognitive assessments. At terminal, we looked again at their inflammatory markers and at their microglia, and we also performed that same spatial transcriptomic analysis so that we could then relate our data back to what we saw in our human patients to better understand translationally relevant targets that we could interrogate further. So in terms of their cognition, what did we see? Well, we didn't really see anything terribly interesting in terms of our water maze analysis, although it did indicate that the high-fat diet animals had a slower learning curve compared to the standard diet mice. We did see some interesting things when we looked at something called puzzle box, which is essentially a task that I'm showing you here on the screen, where we put a series of increasingly difficult objects in the way of these animals so that they have a harder time escaping the light area of this box into the dark area. After all these single obstacles are completed, we then combine them all into one complex obstacle to see if they can remember the previous tasks and combine them to escape the box. You can see here our standard diet mouse, which was what this mouse was, had really no problem with the task at all. The advantage of this particular task is that it is a measure of executive functioning. And if you remember from earlier in my introduction, or earlier in my talk, I mentioned that executive functioning and episodic memory are two of those main learning and memory domains that tend to decrease with increasing age. So what did we see? Well, we actually saw that the high-fat diet mice really had a hard time with this task. And I apologize, I know it's a little bit counterintuitive. I'm showing you some survival curves, and typically a shorter survival means the worst outcome. But in our case, a shorter survival time means that they escaped the box faster. So that's actually our positive outcome. And you can probably tell that our standard diet mice had almost no trouble with this task, whereas our high-fat diet animals really couldn't complete it in many instances. You can also see that reflected here in a slightly different way using hazard ratios. Essentially what that's telling us, if I look at the 12-month time point as an example, is that our high-fat diet animals were 4.68 times less likely to escape the puzzle box as compared to a standard diet counterpart. So they really had a lot of trouble with this task. But what about their inflammatory phenotype? What's going on in their brains? And is this the same as what we saw after 4 days on diet? Essentially, we saw many of the same things in these older animals. Essentially, what we saw was in various different areas of the hippocampus, we saw that those mice are just not able, high-fat diet mice are not able to respond to lipopolysaccharide challenge, whereas our standard diet mice have the expected response with a decrease in that complexity or more amoeboid shape. Well, we didn't see too many differences in terms of comparing our saline mice, so our standard diet mice given saline compared to our high-fat diet mice. We do see this decreased responsiveness to LPS, indicating to us that their microglia are not as functional as are the standard diet microglia. But what about their transcriptomics results? What happens when we look at their brains? What are the pathways and the things that are changing? So we performed that same, as I mentioned, spatial transcriptomic analysis. We identified quite a few more clusters in the brains of our high-fat diet mice, which is to be expected given that we had an entire brain that we could look at versus just the hippocampus. But essentially what we saw when we looked at some of these different clusters that, again, had a high proportion of microglia, that had a high degree of change due to high-fat diet feeding, we found that many of them were located within the hippocampus, and that many of them had a high proportion of glial cells, similar to what we saw in our human samples. And then we performed that same ligand receptor analysis. And what we saw was that, yes, many of these same clusters that had a large degree of change due to high-fat diet feeding also had a large degree in change in signaling. And what does that signaling even look like? If we look at our ligand receptor pair analysis, or if we look at our differentially expressed gene enrichment analysis, we can see, again, we have a high level of change in these neurodegenerative disease pathways or signaling, such as APP or Alzheimer's disease, changes as expected in metabolic or diabetic pathways, and once more, changes in inflammation or immune system signaling or pathways. And many of these pathways were the same that we saw in our human data set. So for example, SPP1 and antigen processing and presentation. which is telling us that our model is recapitulating many of the same changes that we saw in our human sample set. All right, given that we now know that inflammation and immune system changes are present and are likely contributing to this cognitive impairment in our mouse model, our next step was to identify these potential pathways of interest. The one that we started with is an innate inflammatory pathway that's called CGAS STING. And essentially how this pathway works is that it senses intracellular cytosolic double-stranded DNA, which canonically would be from viruses or bacteria. And it works through its different molecules, namely its effector molecule STING and its transcription factors IRF3 and NF-kappa beta, to turn on pro-inflammatory programming, apoptosis, cytokine release, etc. Now, why did we pick this particular pathway? Well, it turns out there's a lot of great data, at least in the periphery, in terms of in adipocytes or in adipose tissue or in liver or in other models, that if you feed animals high-fat diet, or if you create a high-fat diet environment in a dish by giving the cells palmitate, which is a saturated fatty acid, you turn this pathway on. However, if you block CGAS STING or IRF3, you can then block many of these same downstream pro-inflammatory programming that we typically see. So it's blocking some of this inflammatory response that you get from palmitate or from high-fat diet feeding. And in fact, this pathway is really becoming of interest to a lot of researchers in terms of the brain and in terms of neurodegenerative research. Multiple neurodegenerative diseases have been investigating CGAS STING, such as Huntington's disease, ALS, even in AD, ADRD. And in fact, in stroke, which was kind of one of the pioneers looking into CGAS STING in the brain, they've shown that if you block CGAS, you can push microglia to a less inflammatory phenotype, so decrease M1 polarization. You can decrease apoptosis, and importantly, you can even decrease cerebral infarcts in a mouse model. So this pathway is highly implicated in both the peripheral and the central aspects of the body. So our first step was then to, okay, let's understand what might be going on in our model systems in obesity and diabetes in the brain. So in order to do this, our first step was to understand, are we getting cytosolic double-stranded DNA release? So we used a similar paradigm where we created a high-fat diet environment by giving the cells either palmitate, that saturated fatty acid, or palmitate and insulin for 24 hours. And we looked at the cytosol of these cells, both hippocampal neurons and microglia. And we found that, yes, indeed, we do see an increase, or at least a trending increase, in double-stranded DNA in the cytosol in our palmitate-treated cells. So we know that the double-stranded DNA is there. Is it actually turning on CGAS STING? So we did the same experiment, but we looked at these downstream molecules within the CGAS STING pathway, and we found that, indeed, they are increased. In neurons, we saw a tiny increase in CGAS, not anything too exciting. But in our microglia, we saw a really robust increase in CGAS, STING, those transcription factors, phospho-IRF3 and NF-kappa beta, telling us that this pathway is robustly activated under these high-fat diet-like conditions in a dish. That's great that it's activated in a dish, but what about in an animal? Are we actually seeing the same thing in a brain? It turns out that yes, indeed, if we feed our mice high-fat diet for 4 days and we look at their hippocampus, we can see an increase in the high-fat diet-fed mice of CGAS and of STING, telling us that not only in vitro, but in vivo, this pathway is likely turned on and playing a role. Given that information, the next step was to understand, okay, what happens? Is this pathway critical? If we knock it out, what happens to our cognitive phenotype? What happens to potential changes in inflammation and in transcriptomics? So we performed this very longitudinal study where actually instead of starting mice at 6 weeks of age, so as teenagers, we started them on high-fat diet feeding, this time at a year old. So they're already middle-aged when we start them on our high-fat diet. And we fed them for either 32.5 weeks or for 52 weeks, so for a full year. And we again repeatedly assessed their metabolic phenotype and their cognition. And we have some terminal inflammatory parameters, including microglial morphology and some single-cell transcriptomics. Now, these data are currently being processed, so I won't have them all to share with you today. I'm going to share a little bit about our metabolic outcomes and our cognitive assessments. Hopefully, we'll have that full data set to share coming soon. But what did we really see? Well, in this particular data set, one of the more interesting things that we saw, along with just aging-related changes like body weight loss due to age or mortality due to age, we also saw some interesting things in terms of sex differences, because this experiment, we used both male and female mice. Now, I'll preface this a little bit by saying that male and female mice have very different responses to high-fat diet feeding in terms of their metabolic responses. What we tend to see is that in female black 6 mice, while they do eventually gain weight on a high-fat diet, this is delayed compared to males. And this is also true for glucose intolerance. And we've more or less saw this recapitulated in our data set. Again, it's a little bit harder to see because of the loss of body weight just due to age. But the high-fat diet guys, the males are nicely increased in their body weight and clustered here, whereas the females kind of have a bit of a bigger spread. But when we look at our knockout mice, we can see that the female knockout mice gained weight almost immediately and quite significantly as compared to the black 6 mice, the non-transgenic animals. And this was similar for our glucose tolerance test. So you can see the males on high-fat diet, the non-transgenic males had glucose intolerance pretty much right away, whereas the females were somewhat protected from this. But if we look at our CGAS knockouts, both the males and the females developed glucose intolerance pretty quickly after starting on high-fat diet feeding. Turning to our cognitive effects of CGAS knockout and of high-fat diet feeding, again, we saw just a general decrease due to age over time in terms of their puzzle box responses. We also saw, as expected, that the high-fat diet guys were worse performers compared to our standard diet mice. But again, we saw an interesting sexual dimorphism where our CGAS knockout female mice were our worst performers as compared to any of our other groups, indicating that this may be detrimental in the females and potentially somewhat protective in the males, although we are still trying to understand these sex-specific effects. All right, so I hope I've convinced you that we're seeing some very early changes in terms of our pro-inflammatory responses in our mouse model after even only 4 days on diet, and that we have these persistent cognitive deficits that show up in our animal models, along with similar transcriptomic changes in their brains to what we saw in our human data set. Not only that, but that CGAS does appear to be playing somewhat of a role with an acute hippocampal activation in the brains of high-fat diet-fed mice, and that we have some sex-specific effects of knockout that we really need to better understand. All right, next I'll talk a little bit about where I really want to take these data, especially into my new lab, and some brief conclusions before opening it up for questions. All right, as I mentioned, as we've been going through our transcriptomic data sets, one of the focuses of that data and understanding that data has been to look at cell-cell communication. So how are different cell types in the brain communicating to promote neuroinflammation, to promote neurodegeneration? Well, one of the ways we think they might be doing this is through extracellular vesicles, or EVs. Now, these are tiny little particles that all cells release, even under homeostatic conditions, to use to signal to other cells. It's been shown under different disease states that they can contain pathologic cargo that can then, quote unquote, infect cells. And so we wanted to understand, using our paradigm, if these extracellular vesicles might be playing a role, in particular to promote inflammation in neighboring cells. So our first step into doing this was to take our human microglial cell line and to again use those high-fat diet-like conditions and to incubate them with palmitate, and then to isolate extracellular vesicles from the media of these cells. Now, when we do this, what we see is that if we look at this marker, this TSG101, which is a marker for extracellular vesicles, there's actually an increase in the production of these EVs in cells treated with palmitate. And then if we take these extracellular vesicles and we look at their content, we can see that they have pro-inflammatory content in the form of TNF-alpha, and they have cargo that is double-stranded DNA. So in theory, this double-stranded DNA in recipient cells could then activate CGAS sting. But not only that, we wanted to understand what these extracellular vesicles might be doing in naive cells or in neighboring cells. So we performed that same experiment where we took those EVs from treated cells, but we then treated naive cells with those EVs to see what might be happening downstream of that. as I mentioned, in the recipient cells. And what we see again is that, yes, you get that same increase in output of extracellular vesicles, but in our treated cells, in our treated microglia, there is a really strong increase or upregulation of this inflammatory transcription factor, NF-kappa beta, telling us that we are turning on pro-inflammatory pathway in recipient cells. So in order to really dig deeper into this, our next experiments will be based around both in vitro and in vivo models, where we're hoping to dissect this extracellular vesicle-mediated inflammatory crosstalk by doing things like blocking sting in vitro to see if we have that same inflammatory spread in recipient cells. In this case, we're really interested in treating neurons versus treating microglia. Also, we'd like to really dive deeper into the content of these extracellular vesicles to understand what might be driving these changes. In vivo, we're hoping to take some CGAS microglial-specific knockout mice, which we're currently in the process of breeding and have currently on some studies, and to take the microglia from these animals so that we can harvest microglial-specific extracellular vesicles. Using those microglial-specific extracellular vesicles, we will then inject them into standard diet-fed mice to see if that has any impact on cognition, any impact on tissue inflammation or neuroinflammation in the recipient animals. So looking at this both in vitro and in vivo. The other line of research that I will also be pursuing in my laboratory will be centered around this whole idea of obesity and diabetes promoting early aging. Now, we know that aging has multiple effects in the body, multiple cellular effects, including things like telomere attrition, mitochondrial dysfunction, cellular senescence. It's in fact been shown that in obesity and diabetes, many of these same effects are present, such as it's been shown that it causes cellular senescence, changes in intracellular communication, and it's also been implicated or hypothesized to have effects on things like stem cells, telomeres, epigenetics, etc. So all of these same effects of aging are potentially present in obesity and diabetes. And one of the ones that we're particularly interested in exploring is senescence. And why are we interested in this? It's because in senescent cells, not only do you have changes in cell function, changes in cell morphology, you also have these cells releasing something called SAS factors, or senescence-associated secretory phenotype. And this contains many pro-inflammatory molecules such as IL-15, IL-6, activin A, etc., that can then promote senescence in neighboring cells. So again, kind of harking back to this idea of cell-to-cell communication that's promoting pathological changes in the brain. And one of the reasons we think that this might be happening in our models is using some of this new data that we have from the single-cell transcriptomics, where we looked in our 2-year-old mice that had been fed diet for an entire year. And what we see is that we can identify many of the expected cell types, such as endothelial cells, neurons, oligodendrocytes, astrocytes, T cells, and we have a very large microglial population. But then when we take this data set and we apply a very specific bioinformatics program that's intended to identify senescent cell signatures. So in other words, to allow us to identify which cells are becoming senescent, we see that this transcriptomic signature is really showing up mostly in our microglia. So again, it's telling us that possibly this chronic microglial activation that we saw in both acute and our chronic models might be causing senescence of these cell types, which might be contributing then to cognitive impairment and to disease. So in terms of our experiments that we're hoping to pursue to understand the underlying mechanisms of all this, in terms of the senescence side of things, we're hoping to use a senolytic, which is an agent that causes the death of senescent cells called fisetin. We're also hoping to use some just lifestyle interventions, such as exercise, which has been known to have beneficial effects in terms of reducing senescent cell burden. We're also starting up some new collaborations with some heart researchers because we know that both the heart and the brain can be impacted in obesity and diabetes. So we have a couple of ongoing collaborations where we're trying to understand underlying immune or inflammatory changes in both the heart and the brain and how those two organs might be communicating to potentially promote disease as we're aging. All right. So just to sum everything up, we saw from our clinical data that yes, indeed, obesity and metabolic dysfunction are promoting cognitive impairment. In particular, we saw those changes in episodic memory in that Pima Indian cohort. When we look at our data, we see from our transcriptomics data in our human patients, we see not only changes as expected in neurodegenerative and metabolic pathways, but really also in immune or inflammatory pathways and signaling that are really highly concentrated in the microglia cells or in glial cells. When we took those data back into the bench, we saw metabolic, immune, and pro-inflammatory changes short-term that we also saw in our longer-term experiment, which was in tandem with our persistent cognitive impairment. I hope I've also convinced you that CGAS sting is indeed playing a role in this with the acute microglial activation in vitro as well as in the hippocampus in our mouse models, and that we have some really cool, potentially interesting sex-specific effects that we're trying to explore. And then finally, my future directions will then center on inflammatory spread or crosstalk, not only between cells within the brain, but also between organ systems, namely between the heart and the brain. All right, with that, I really want to thank everyone for your kind attention. I'd like to also thank, of course, my mentor, Dr. Eva Feldman, and the members of the Feldman Lab, who are all amazing. So many of these studies, as you can imagine, feeding mice for a year is no small feat. Tons of great technical help in the Feldman Laboratory, particularly help from Crystal Packett, John Hayes, Diana Regan. I also get a lot of help and support from my other fellows. So from Stephanie Ayed, who is now actually a research faculty here in the department, and from Benjamin Murdoch. I, of course, have to thank our statistical partners, both Lily Zhao and Ali Turfa, for helping to analyze that longitudinal cognitive data, and Jung Akur and Kai Go, who are our bioinformatics partners. I also really want to take the time to thank Jeff Murphy and the Murphy Lab. They were the ones that got my feet wet in terms of all of these behavioral analyses. I spent a lot of time with Jeff and his lab learning how to assess cognition in mice. And then, of course, can't forget my funding sources, the T32 training program that got me off the ground here at Michigan, the MADRC Early Career Investigator Mentorship Program, which I was fortunate enough to be part of and helped me obtain the human brain samples to do the spatial and the single cell analyses, the Edith Brishkin Emerging Scholars Program, and of course my NIA K99. And I'd be happy to take any questions.
Great job, Sarah. And I wanted to start by saying that I'm happy you're joining MSU soon. So looking forward to have more conversation with you. Before I continue, I want to acknowledge that Stephanie did a great job taking people out of the Zoom that were trying to disrupt the meeting.
Thanks.
I know they always try to use racial sensitive issues to offset people. So it's something that we hope we had the control of it, but some people spend the time in a weird way. So I want to acknowledge how sensitive that could be to some of the people in the call. So I hope that in the future we can talk about proteomics and how we can collaborate in the future to enhance your transcriptomic data, but there are some limitations, especially when you look at receptors and signal molecules, and perhaps proteomics might not capture that and transcriptomic may. So there's some crosstalk between transcriptomic and proteomics is very difficult, but it's because it's analyzed out of context. So I hope we have that conversation in the future. But my question is, it's always very interesting that high-fat diet, especially with the animal models that express amyloid or tau, exacerbate the pathology. But in your case, it seems that you see all this cognitive impairment without the aggregation or any pathological hallmark. If we go to human, could it be possible that diabetes and metabolic disorder prime our immune system? Because we have people that develop pathology without ever presenting symptoms, right? So maybe it could be a parallel rather than a cross-talking, phenomenon. What do you think about that?
Yeah, I think that's a really great point. Thank you for that. That's part of the reason why I'm really interested in understanding changes as disease is progressing, right? Because it's hard to kind of dissect whether or not things are happening in tandem, or whether you're getting the crosstalk, or whether you're getting, one promoting the other. And I think that's something that I'd really love to understand a lot better. Because immune system changes are typically thought of as reactive, but I think more and more people are recognizing that it's potentially causative, not so much reacting to the pathology. But that's just such a big area of research that nobody really knows yet. So I'm excited to kind of understand those mechanisms.
Okay, we did have a question in the chat. It's from Lisa Torres, and it says, if the high-fat diet mice change to a low-fat diet, does that correspond to improvements in the brain and ability to complete complex tasks? Can the damage be reversed?
Yeah, that's another excellent question. Thank you. So yes and potentially no. So we've done a few experiments. Eva had a fantastic ex-postdoc, Katrina Sims Robinson, who did some of that early work where she looked at high-fat diet feeding, and then she put them back on a standard diet and so-called dietary reversal. And she did see an improvement in Morris water maze outcomes. However, we've subsequently done some, and I don't want to give away too much, where we find that actually exercise is more beneficial in terms of reversing some of these cognitive deficits than is dietary reversal or changing the diet. So it's possible that some of the cognitive deficits are reversible, and it depends on the method in which you use to kind of combat that, whether it be diet, exercise, combination of the two, et cetera. So that is something that's also an area of research that I'd like to look into. As I mentioned, the whole exercise piece, I do think exercise is going to be more beneficial, or perhaps most beneficial in combination.
Right, another question in the chat from Anna Doherty. Are the particular vulnerability of specific subfields in the hippocampus that you see?
Yeah, so we've assessed, as you probably some people very astutely noted, we always assess different regions of the hippocampus. So we look at the hilus, we look at the molecular layer, and the CA1, and there are some differences in terms of the microglial morphology of those different regions. It does appear that it's somewhat consistent in terms of you get that inability to respond to LPS regardless of what region you're looking at. But in terms of how activated the microglia are, there is some variation. We're not able to quite get down to the resolution in the spatial transcriptomics to understand changes at that level in the mouse brain. But in the human brain, there is a little bit of difference in terms of, that was part of what I showed in the spatial transcriptomics human data. So you're getting that difference in communication, you're getting that difference in how the gene expression is changing between the different regions, but we've not done too much other than the microglial morphology in terms of the different hippocampal regions. So that's something to kind of look at, because that is an interesting area depending on where you are in the hippocampus.
All right, Dave Morgan has a question.
Yeah, the first part of the question is to make certain I saw what I thought I saw, which was that in your gas knockout female mice, you actually produced larger weight gain than you did in the non-knockout mice, the wild type mice. Yeah. Is that correct?
That's correct, yes.
And so one of the things that we've been discussing in the Alzheimer's field is that these microglial and other reactions to atypical, non-survival related phenomena is that there are a combination of things that are both beneficial and detrimental. And is it conceivable that in fact this inflammation that you're seeing with the diet is actually a response to the diet itself and is providing some protection in the females that is eliminated by your knockout?
So that is indeed possible. I don't think it's the case in that instance. I do think in many cases, high-fat diet-induced inflammation, at least acutely, is a protective mechanism. But of course, if you continue to feed the high-fat diet, that's going to become deleterious eventually. That's the hypothesis. But In terms of the knockout females, I don't think that's the case, at least in our data set, because we saw worse cognitive impairment. Like they were our worst performers by far. So I don't think it's protective in that instance, although it could be protective.
But that's kind of the point, is that they became obese and therefore, because they didn't have that protection. And so I'm just saying that these considerations are somewhat important as we go forward as to how we're going to intervene in these processes. The other interesting point is that an extremely high-fat diet actually causes people to lose weight, and that is, of course, the ketogenic approach. And that the diet you're using, I assume, reduce protein in order to increase fat instead of reducing carbohydrate. Is that true?
I believe it reduces both proteins and carbohydrates. I'd have to go back and look at the diet breakdown. But this level of high-fat diet feeding does not put the mice into ketogenesis. We do have other studies currently ongoing with a Ph.D. student in the lab and with another postdoc that are focused on ketogenic diet. And in fact, we think that one of the important considerations for ketogenic diet is the type of fat that you're using to increase the fat content. So a saturated fatty acid-based diet versus a non or a monounsaturated fatty acid diet is going to be the more beneficial, which is more beneficial versus which is deleterious. So you're right, important considerations, definitely.
Thank you.
Right. Anything else for Sarah before we close things up for the day?
Very well done, Sarah. Thank you for representing your work so beautifully.
All right. Well, thank you so much for the talk today, Sarah. And thank you all for sticking around through that little fiasco. And we hope to see you at our next Leaders Initiative seminar. Take care.